Okay, good morning, everyone. So today we're going to be continuing on our discussions talking about anatomy and physiology. So again, just kind of the previous chapter rundown. We started with a brief overview of anatomy and physiology, things like orientation, and just kind of defining what anatomy and physiology are in chapter 1. Chapter 2 was basics of chemistry, atoms, molecules, macromolecules, maybe a little bit more than you had kind of bought into for an A&P course, but you'll see kind of how those things apply to some of the larger structures when we get there. And then chapter 3 was talking about cells, kind of the general cell, not necessarily specialized cells per se, and that's where we're going to kind of take off here. But those cells, the different parts of the cells, we talked about cellular processes, things like exocytosis and endocytosis we discussed in chapter 3 and part 2, and we'll actually kind of see how that material applies to this specific chapter. I would probably make the argument that this chapter almost has more diversity than we'll see in a lot of the other chapters, just because we're talking about tissues. So tissues have a lot of different places that they can be. When we talk about, skeletal structures such as bones, we talk about muscles. We kind of in our minds have a picture of that. When we talk about tissues, there's so many different tissues that can actually take place in the body. So it's actually kind of a very diverse and widely spanning topic. So if we go back to that figure in chapter 1, I should probably just start putting that at the beginning of all of these lectures, is that you have your atoms. Your atoms form molecules, your molecules make macromolecules, your macromolecules make cells, and then your cells come together and can form tissues. So we're just really stepping up our levels of complexity there. So today we'll talk about tissues. In the rundown of things and expectations for the course, please make sure you're staying on top of these things. Make sure you answer the study guide questions for chapter 4, maybe as you're watching this lecture. Quiz 4, chapter 4 is due Friday by midnight, and homework 5 for adaptive practice chapter 5 is due next Monday. Please make sure that you're staying on top of these. Again, maybe one way to remind yourself is just to set an alarm in your phone to make sure that you're not missing these due dates. But I'm hoping now that we've fallen into maybe a rhythm of the semester, this being like the 3rd and 4th week of the semester, that hopefully you're kind of in your mind saying, oh my gosh, it's Friday, I need to make sure I do my quiz, or oh my gosh, it's Monday at 11:50, I need to make sure I do my adaptive practice. So make sure you're staying on top of these things, because those points do add up over the course of the semester. The adaptive practice, is based off of completion. So hopefully that makes it kind of a low stakes type of assignment. And then, at the end of the semester, if you have done all of your adaptive practices, you basically have earned more points from those adaptive practices than any one exam. So they do add up, they do mean something. So make sure you stay on top of those assignments. Okay, this is week 4 that we are looking at here. Just a heads up, week 5, we'll be looking at our first exam coming up. So just kind of keep that in mind. In the back of your mind is that our first exam will be coming up here not too shortly. Okay, so the study guide questions for this, I just like to post this here to do a quick rundown of what we're going to go over today. Also recognize that the study guide questions are posted online as well. These will be the study guide questions that we'll be going over in class together. However, I don't think we'll get through this full list of study guide questions that's shown here. We might not be talking about the properties of various types of membranes. That might be in the following lecture. So just recognize that I'll probably change that on the study guide that's posted to CourseSite. And then we'll probably omit it when we discuss it in class, because it's not, I don't think, included in this lecture here. Okay, so this particular chapter about tissues. So we're going to learn about tissues and their origins, discuss how tissues are held together, talk about epithelial, connective, muscular, and nervous tissue. In this particular lecture, we'll focus on epithelial and connective tissues, and in the second lecture, or chapter 4, part 2, we'll talk about muscular and nervous tissue. Those two tissues we will revisit later when we're going over the specific chapters. So this chapter is kind of just a basic introduction. We'll talk much more specifically about those tissues in those particular chapters. Learn about the structure and function of membranes and understand tissue repair, because as tissue is damaged, it has to be replaced and repaired. That's one of the marvelous things about our bodies, is that we can repair ourselves most of the time, not all of the time. Some tissues are more readily repaired than others, given kind of the turnover of those cells. Okay, so types of tissues, very broad introduction of the different types of tissues that we kind of just talked about. So what is a tissue? Probably the best place to start is with that definition. So it's a a collection of cells that have a very common embryonic origin and carry out a very specialized function. So if you think of post-fertilization, very early on in the process of becoming a human, you have these cells, these stem cells that get differentiated or change into a cell type of a very specific type. So in the case of a tissue has maybe a single cell that actually originates, and then once that cell starts to divide, it makes more and more of the same type of cell that has one specialized function. I talked about this in chapter 3, and I think it's worth reiterating here, is that all of the cells in your body carry the exact same DNA, the exact same DNA sequence. And the only thing that makes those cells different from one another is the way that that DNA is read and expressed. And so they all have the same basic blueprint, but they can express themselves to be very different types of cells, very different types of tissue. So if you think of your skeletal tissue versus, the tissue that's in lining your stomach, those two tissues have the same DNA. Just the way that they're expressed makes them very different in their structure and function. What's interesting also to note about that, maybe if you're interested, is that because all cells have have that DNA, some research labs are starting to investigate the idea of cellular reprogramming. So taking that programming that cells have to be one tissue type and reprogramming them so they'll become a different tissue type. So if you are missing one type of tissue and you need to replace it, that's one way in which researchers are starting to investigate. So there's some cool science there that's happening that maybe in the next 10 to 20 years we'll actually see applied in a physician's office, but I think we're kind of a ways away from that. Okay, so there are 4 basic tissue types in the human body that we saw in the previous slide, and then I think the next slide is going to show us images of those and our categorized according to their structure and function. In an A&P class, I'll just tell you right now, just get used to hearing the term structure and function like it's nothing, because we're just going to talk about structure and function again and again and again and again, just because that's so much of what anatomy and physiology is, right? Anatomy is structure and physiology is really the function. So it just kind of is reiterating the title of the course. So the types of tissues that we see here, we have epithelial, connective, muscular, and nervous. So these are the 4 different types of structure. Again, they have common embryonic origins, meaning they have a single cell that is derived and differentiates into that cell type, and becomes like all of the epithelial tissue. Another cell becomes all of the connective, all the muscular, and nervous. So here we can see epithelial tissue, how it kind of structurally looks different than the other types of tissue. You can see that there's kind of like sides to that tissue, and we'll talk about the different sides. So this side is kind of the bottom or the basal membrane. We'll talk about that terminology. And the other side you can see here has all of these little fingers on it. Remember last chapter we talked about flagella and cilia. These are a bunch of cilia up here, and those are exposed to the outside. I should say outside world, but they're exposed to the outside and are interacting with the outside. They line a lot of our membranes and organs and can assist in the movement of fluids. And you can kind of see the structure up here. It's a single layered structure most of the time, and then you can also see the shape of those cells. And so arrangement and shape, we'll revisit that idea multiple times over the course of this lecture, because those are kind of the ways in which tissues are characterized, is by the arrangement of them, meaning are they single layered or multi-layered, and then the shape, and then we'll go over the shapes here in a second, but I just kind of wanted to start planting that seed now. Again, epithelial tissue has these different sides that we'll talk about, and lines membranes. Connective tissue, next you'll see. Here kind of is almost what it sounds like, connective tissue. It's joining together things like organs. You can see here how it's vascularized. You've got this blood vessel that's going through it. And so when we talk about things being vascularized, it means that there are blood vessels that are bringing blood to the region. If it's lacking blood vessels, it's considered avascular. And so epithelial tissues would be considered a type of tissue that is avascular. Vascular. And then connective tissue is vascularized. Vascularized. Okay, meaning it's got blood vessels bringing oxygen to those tissues. We'll talk more about where connective tissue is located. Here it's interesting, you can see the vascularization. You can also see that there are some fat cells here. Connective tissue kind of houses energy as well. And again, it's kind of the glue that holds different parts of our bodies together. Now in this lecture, we won't talk about muscular tissue. We'll talk about that more in part 2, and then we'll also talk about it when we get to the chapter talking about muscle structure. But here you can see kind of the nice shape of muscle cells, the different mitochondria. I believe that's what's being represented here. Just because your muscle cells require so much energy, they have a ton of mitochondria within them. They are long, kind of cylindrical structured cells, and again, the main function of those is going to be to create force and tension against our skeletal to create movement, so to create kinetic movement. Lastly, you can see nervous tissue here, a nervous cell. We'll talk extensively about this, and I'm going to try to not lose myself talking about every part of the nervous tissue because, again, it's kind of my thing, but here you can see all of these extensions out into the cellular environment. One of the main functions of nervous tissue is to send and receive signals. And so because of that, the structure of nerve cells kind of lend itself to that. So all of these projections that you see out here are going to be receiving signals. And then once it receives signals, it's going to send its signals down this long kind of tail structure called an axon. We'll talk about that more when we get there. But again, one of the main functions of nervous tissue is to send and receive signals. So again, lending to that whole structure function idea, you can see here that each one of these tissues has very different structures, and because they each have their own specialized function within the body. So epithelial, connective, and muscular tissue are the 4 types of tissues. Okay, so cell junction. So because we're kind of making the transitions to talking about singular cells, what the functions of singular cells are, I think in chapter 3, I hit home pretty hard that cells functions are to be little protein making factories. But those proteins that they make are going to dictate what functions they have and going to also dictate their structures. And so we're going to see how some structural entities are what allow tissue types to actually have their structure. So for cell junctions, what we're talking about here is how these individual cells are basically glued together to create those larger tissue structures, right? Because we're not talking about single individual cells kind of floating in the environment. They're all kind of tightly woven together. Maybe woven is not the best room, but they're kind of tightly stuck together. So we'll talk about how those cell junctions, what they're actually composed of. So they can be held together, as the slide says, in a number of ways. And these are all points of contact between neighboring cells. And so basically it's almost like you are, if you are an individual cell and you're trying to link, kind of trying to create a tissue or of a group of people, what you would do is kind of link your arms together. And so cells kind of do the same thing, except for they create probably more permanent interactions with other structures. You can also think of them as like patches on a quilt. There are different ways in which you can connect those patches on a quilt to create kind of one continuous structure that isn't, able for things to kind of pass readily or easily through. So we'll look at kind of the example that we're looking at here is of epithelial tissue. Again, I know that because I can see those cilia on the top, and I can see the basement membrane down here, which is at the bottom. And so those two sides are apical and basal. And what's nice is that these epithelial cells have all of these different type of junctions that we're going to talk about here in a second. Okay, so intracellular junction interactions animation. If you are watching your own PowerPoint, or if you're going to open the PowerPoint, you can click on this link here and it'll send you to a video on Wiley Plus. Many of these videos are really nice supplements to what's we're discussing lecture. And the thing about it is a lot of the processes that we're talking about are exactly that, they're processes. And so seeing those processes happen in real time through a video can be really advantageous. So I do not take it personally if we're discussing something in lecture lecture and then you go to YouTube or you go to this video and you watch the actual process happen, because it gives you a really wonderful visual of what's happening as it's happening in the cell. So please make sure you're utilizing that, especially if you're a visual learner. I think videos and it can be really helpful for the visual learner. Like I said, I'm super bummed this semester that we aren't able to meet live for a variety of reasons, but probably one of my biggest downside is the fact that I love virtual reality, and man, are there some cool virtual reality apps for anatomy and physiology, but maybe sometime in the future. Okay, so back to class. So in terms of these types of junctions, here you can see the first type of junction, which is called a tight junction. You can see that this tight junction this is a plasma membrane of one cell, this is the plasma membrane of another cell, and you basically have this intracellular space that exists between them. Now, if you have a whole line of epithelial cells, you do not want extracellular fluid just leaking between these cells really easily, right? You want to have a very tight connection between the two. So you've got these strands of transmembrane proteins you can see here that kind of look like beads that are going to help kind of weave the two extracellular membranes of these two cells together. So these two adjacent membranes allow for essentially fluids not to leak through those super tight areas between the cells. So exactly what it sounds like, tight junctions are exactly what they sound like. They're very tightly woven areas between two cells that are basically anchored together with these transmembrane proteins. If we again, I talked about protein production, how important it is for cells. Transmembrane proteins mean that they span the entire extracellular membrane of the cell. So these transmembrane proteins are able to anchor all the way through the cell membrane into the other side of the cell. So tight junctions, exactly what it sounds like, are tight and don't allow fluid to kind of leak between that one single layer of epithelial tissue. Adherence junctions are another type of junction, so we don't just have tight junctions, you can also have adherent junctions. Similarly, you can see that there are these adjacent plasma membranes, so you've got essentially cell 1 over here, cell 2 over here, this is plasma membrane. You can see that there is this plaque, again it's formed of proteins that line the inside of the cell, and embedded into that plaque are going to be these transmembrane glycoproteins. If you remember what glycoproteins are, essentially proteins that have sugars attached to them. And so these sugars essentially interact with one another and anchor them to the cell membrane. So those sugars have very specific structures that identify one another. And so you can kind of not create a lock and key, but kind of like a lock and lock, where they kind of come together, create these very, I shouldn't say tight junctions, but these very adherent junctions and allow for these two cells to essentially adhere to one another between the intracellular space. And so not as like kind of continuously interacted as tight junctions. Adherent junctions kind of occur in specific locations, but again, they're very similar to the same function of holding those epithelial cells together. And these don't occur just in epithelial cells. We're using epithelial cells just as an example here because they have each one of these junctions, but you will see these types of junctions in other types of cells. Desmosomes are another type of essentially junction. You can see here that these desmosomes also have these transmembrane proteins, these glycoproteins. In the case of desmosomes, that glycoprotein is called cadherin. Adhesive cadherin, and then you also have a plaque that's the base of where that cadherin is anchored. What's interesting here is that you also have this intermediate filament or keratin that is interacting with the plaque. And what this is going to do is it essentially allows this junction or this interaction to not just be anchored to the plasma membrane at a specific location, but you're also kind of interacting with those tadpoles of the cell as well. So it's like a deeper anchor because you're interacting with the anchors of the cell. So if there's a structural change or a movement that occurs within that cell that would affect the the intermediate filaments, that's going to affect, because it's anchored to other intermediate filaments of the cell next to it, it's going to hopefully alter the larger movements of that cell next to it. So again, it's just kind of a deeper anchoring of the cell. The interesting thing to note about desmosomes is you have these, the cadherin plaque and an intermediate filament kind of structure on both sides of them. We're going to talk about the other example here in a second. So you've got in cell 1 and cell 2, you've got that cadherin plaque intermediate filament interaction on both sides. The intracellular space, when I say intracellular, I'm just talking about the space between those two cells. Okay, so desmosomes, also important interacting points in those epithelial cells. Okay, so to contrast desmosomes to hemidesmosomes, hemi, almost like meaning half. Here you can see you have a desmosome that's located on one side of the cell, like we just talked about with the cadherins, plaques, and intermediate filaments. That intermediate filament specifically is called keratin. But what's interesting here is you've got the cell, let's say just like cell 1, you have the bottom of cell 1, and it's adhering to what's called a basement membrane. The basement membrane is going to be the essentially bottom of the cell. If we talk about the different nomenclature for an epithelial cell, this is also called the basal side of the cell. And we'll talk about the other side here in a second. I don't want to introduce too many ideas in this one slide. But you have your basal side that is being adhered to the basement membrane. And you can also see that this transmembrane glycoprotein, in this case, it's integrin, not cadherin, that's occurring in the extracellular space. And this hemidesmosome, you only have the presence of a desmosome on one side because you're adhering to a basement membrane, which is a different type of tissue. Okay, gap junctions. So we're rolling our way through these different types of junctions and interactions. So gap junction, you've got, again, cell 1, cell 2, I should probably write it to you there, cell 2, and you've got this this gap between them. And what gap junctions do is they kind of create this like little signaling area between those two cells. So the way that the signal passes is through these proteins called connexins. Connexins are transmembrane proteins, meaning they span the entire membrane, and they create a essentially kind of like a portal or a hole in which things can pass between those two membranes. Generally, what gets passed between them can sometimes be cations or anions, which can be very helpful for signaling. So gap junctions, because of the ionic movement or ions and other types of molecules can move between the gap junctions. They can make them a very fast signaling mechanism. And so there are places in the nervous system where gap junctions are used. It's also used very early in embryogenesis when you're trying to, again, send very, very, very quick signals, because neurotransmission itself is very quick, but with an action potential occurring, again, that's kind of I don't want to say longer, because it really is very quick, but if it needs to be almost instantaneous, these gap junctions occur. And so this can also help mediate coordinated movement of an entire tissue, let's say a contraction, specific types of tissues that allows that to happen instantaneously in a coordinated manner. So gap junctions, again, form these kind of hole portals with these different proteins that span the transmembranes and allows ions to move through. These transmembrane proteins here are called connexins. They connect the two cells. Hopefully that kind of, nomenclature you respond to connects the two cells, and again allows just easy passage of cations, anions, or other molecules between the cells if they need to pass through really quickly, if they're connected really tightly. Okay, so again, those are different types of junctions, kind of how the cells are kind of interwoven together. And like I said, we'll probably focus primarily on connective and epithelial tissue in this particular lecture. So let's go ahead and compare the difference between epithelial and connective tissues. So right here, quick pause. What differences can you see between this epithelial and connective tissue? So I'm just literally going to pause and see if you can maybe talk about, think about how they're different. Okay, so probably the most striking difference between these two types of tissues is essentially like the number of cells and how tightly packed those cells are, right? So epithelial tissue has many cells. You can see many cells. Each one of these little purple dots here is actually a nucleus. So each nucleus that's present here essentially tells you that there is a cell present. So this is just a type of dye that people use to dye nuclei. So every place you see a nucleus, there's a cell. And they're packed together with little to no extracellular matrix. You'll also recognize you don't see any kind of blood vessel. This is avascular, as we talked about earlier, so you don't have vascularization going to this epithelial tissue the same way you do connective tissue. If we look at this connective tissue, you can see that there are far fewer cells. You do see some vascularization that might be indicated by these purple lines. So there's blood vessels that are occurring. So that's going to be kind of the main two differences between epithelial and connective will be the number of cells and the way that they're arranged, as well as the avascular versus vascularized state of them. Although I mean, as a scientist, I probably would make the argument that it looks like these two figures are on different scales because look at how large the nucleuses are in the epithelial tissue versus the connective. So I don't know. If I was a textbook editor, I would have made these the same scale so you can see exactly comparatively like the difference between, because I'm guessing connective tissues nuclei aren't like 5 times larger. No, that's not the case. So scale, man. Okay, so next let's talk about epithelial tissue and some of the different structures, cell types, things like that. So as we saw in the previous figure, they're arranged in sheets and are very densely packed. Many cell junctions are present. Again, when we were talking about cell junctions just now, that was the type of tissue we were using as an example because all of those types of junctions are present in epithelial tissues. They attach to a basement membrane. So before we were looking at the differences in the kind of sides to the epithelial tissue cell, and we'll go over exactly what those sides are called and how they're attached to the basement membrane. And epithelial tissue is avascular, meaning that it does not have active blood supply or vascularization to it, but does have a nerve supply, meaning that there are nerves that are located within epithelial tissue. So in any type of tissue that you're talking about, those are usually kind of the two things that you're looking for is vascularization and nerve innervation, telling you is there being blood and oxygen being supplied to that particular tissue, and then nerve supply, is there a way for your brain to essentially send and receive messages to that particular region? Mitosis occurs frequently. We talked about that in the last lecture of chapter 3, mitosis being cell division. And so since these cells are not necessarily cancerous, this is a very tightly regulated process. But because they are dividing so quickly, these can be regions that can become cancerous because mitosis is occurring if that it becomes unregulated in some way. But mitosis is occurring because you've got sloughing or loss or death of these cells, and those cells have to be replaced in some manner. So that's why mitosis is occurring, because those cells are being lost in some way for those tissues. So the surfaces, I got to erase. So the surfaces of the cells and the basement membrane, this is a nice figure showing that, kind of been alluding this multiple times. So again, what we have gone over is I introduced the idea of the basal surface that is attached to the basal lamina. Underneath the basal lamina is the reticular lamina, and these are both considered the basement membrane. That's what those cells are anchored to. Remember, we talked about how hemidesmosomes are the way in which the epithelial cells or epithelium is anchored to that basement membrane. You can see here that the lateral surfaces of the cells are those surfaces between them. This is where things like gap junctions and desmosomes and tight junctions can occur between these lateral surfaces. So there's no type of leaking between those cells, as well as there can be communication between neighboring cells as well. Okay, so I kind of lost sight of what we were originally going over, which is the annotation of the basal side. The cell opposite of the basal side is the apical side. So the apical side is a free surface side that is exposed to the extracellular environment around it. This is most likely where the interaction of the cells are. So I always think of these cells like when I'm thinking about epithelial cells, I always think of like lining of our intestines, because often, you have this area in which these cells are absorbing nutrients from our bodies. That's how our nutrients are absorbed, so we can metabolize them. And so you've got all your foodstuffs that are broken down that are passing by it, and these, ciliated epithelial cells are absorbing those nutrients. And so it's the apical side of those cells that have those cilia, and it is the basal side that is attached to the basement membrane. And again, if you're having a hard time remembering the nomenclature, just do, again, things like letters. Basal, basement, right? Basal, basement, both start with B. Those two belong together, and then your apical side is exposed to the surface. So as long as you can remember basal and apical, hopefully by process of elimination, you can remember which one is in the bottom basal, basement. Okay, underneath that basement membrane, again, we'll talk about membranes a little bit more in the next lecture. You have your connective tissue, and you can see things that through the connective tissue are things like your nerves that are innervating your epithelial cells, as well as the vascularization of the blood vessel here. And so that's going to be one way in which oxygen reaches your epithelium, is by passing it from the connective tissue underneath. So we're going over epithelial tissue, but we're also kind of getting introduced as to what some of the functions of connective tissue are as well. So not too far away from the epithelial tissue is going to be the connective tissue. Okay, so classification of epithelial tissue. So there are ways in which we categorize epithelial tissue based off of two different characteristics. First, you have the arrangement of the layers, simple, pseudostratified, or stratified. Simple, meaning that there's just basically one layer. It's very simple, just one single layer of tissue that's lining a surface. Next, you can have pseudostratified, which you can see here looks kind of like simple, but the structure of the cells are a little bit different, and they're not necessarily all the same size. Some of the cells look like they're more on top, some of the cells look like they're more on bottom, but they're not kind of equally exposed to both the basal and apical side. Next, you can see in a stratified epithelial tissue, you've got multiple layers of cells there. So it's not just that single layer, you've got multiple layers of cells built upon one another. So these are all types of arrangements of cell layers. Okay, so that's one way you can characterize epithelial tissue. The other way is by cell shape. And so cell shape, you can see here is squamous, which the examples that they're giving you here are almost all simple. They're kind of a squarish shape, but they're very flat. You see that there's not a whole lot of three-dimensional rise on them, versus kind of the second type of cell shape, which is cuboidal, which hopefully that makes sense why they're called cuboidal, because they're raised and they look much like a cube. And then next you can see that the next cell shape is columnar, and again, hopefully this nomenclature makes sense to you, is that those cells are shaped very much like a column would be shaped. So arrangement of layers and cell shape. These are the two ways in which epithelial cells can be classified, meaning that you can have a combination of them. So you could have simple squamous, simple cuboidal, or simple columnar. So those are the two kind of ways. It's almost like if you're talking about hair, right? You can have brown curly hair, brown straight hair. It's very similar kind of in that way. Or you can have, black straight hair, black curly hair. It's a combination of two different characteristics to describe these cells. So it describes the shape of the cells and then how many layers thick they are. So because of this combination, it starts to these types of cells can accumulate very quickly. And so I think the next the table. So this table 4.1 in your book is, I have the book textbook right here, is how many pages is this? 1, 2, 3, 4, 5, 6, 7. So 7 pages in your textbook going over just these different types of tissues. Now, I would recommend just taking a glance over these different types of tissues and looking at things like simple squamous and just, again, looking at some examples of these different types of tissues. We're not going to go over them here in class. I feel like we would get lost really quickly and you probably would fall asleep, to be quite honest, because it's, it's the same thing repeated again and again, like simple squamous, simple cuboidal, non-seal, it's simple. But definitely go to your text. If you're on Wiley Plus, then you have access to the e-text, and you can look at all of the different cell shapes and arrangements, and then examples of each, which I think is really nice. And I think when you do that, you really get a sense of how diverse epithelial tissue is and how many different regions of your body you find epithelial tissue. So it's pretty amazing. Again, I think when I introduced this chapter, I said, chapter 4 might almost be not one of the hardest, but just one of the most diverse in terms of the cells and the different functions that those cells have, because they're, you're covering all of the different regions of the body. So with that being said, please open up this PowerPoint, click onto this link, watch this video. I think it'll give you kind of a good sense and introduction to all of these different tissues. So epithelial naming combinations. So like I said before, the combinations accrue more quickly than you can imagine. So you can have simple squamous, simple cuboidal, simple columnar. Pseudostratified tend to just be columnar. So you kind of get out of the squamous and cuboidal nomenclature there. And then stratified, you can have squamous, cuboidal, and columnar. So those combinations, again, add up very quickly, but in each one of those, the structure lends itself to the different function. And if you go through the table in your book, you'll see some of those different functions. Okay, so next we'll go over different types of glandular epithelium. So it's a single or massive cells adapted for secretion. So I hope to kind of like lay the foundation of secretion in chapter 3 when we're talking about things like exocytosis, but that's not the only way in which secretion occurs. So we'll talk about how secretion can occur for endocrine versus exocrine. So endocrine glands enter the interstitial fluid and diffuse into the bloodstream. And so when you are learning about the human body, there are different advantages or different tools that the body uses to signal. So the nervous system is really good at fast, rapid, immediate signaling that lasts. In most instances, it's pretty transient. It occurs pretty quickly. If you need like long-term signaling that occurs over an extended period of time, endocrine signaling can be far more advantageous. Also, the advantage to endocrine signaling is that you can go across a large distance in the body as well. Not that you can't do that with nervous system, but endocrine, again, is a slightly different mechanism by which signaling can occur. So the endocrine glands release things like hormones, and they diffuse into the bloodstream, and they can, diffuse and send these longer term signals to different regions of the body. Conversely, from endocrine, you have exocrine glands, which enter ducts that empty into the surface of a covering or lining. So we'll talk about some examples of where these secretions are found, maybe in the digestive region, maybe on the surface of your skin. So since skin surface and lumen of the stomach are the 2 examples they give here, so we'll talk about how that happens for exocrine glands and kind of what the different structure of those glands can be as well, because not all glands look the same. Okay, so endocrine glands, like we said, secrete hormones. Or the advantage of hormones is that they're a signaling molecule and diffuse and flow through a duct. They'll just be described far more in depth in chapter 18. We'll hopefully get there-ish. But the pituitary gland is a great example of a gland that's located in the brain. The pineal gland, the thyroid, and these are all glands that are found within your body that send these long-term signals that mediate things like metabolism or just, growth, things like that. adrenal glands to the kidneys, and the pancreas near the stomach. These hormones regulate many metabolic and physiological activities that maintain homeostasis. Again, because these are much longer signaling molecules, those hormones exist kind of in that bloodstream far longer. So if we look at the example here, we can see the thyroid gland that's kind of located at the base of your larynx. And you can see some of the different shapes of the cells that are given there. So you have these hormone producing epithelial cell. Those are cuboidal cells if we're going to talk about shapes, right? Those are simple cuboidals that are lining that area. So you can see next to that they are avascular, but it's not through the tissue itself. It's right underneath. You've got a blood vessel, but a thyroid follicle that's going to be actually releasing that particular hormone into the bloodstream. So it's kind of how we've got the access to the bloodstream for these endocrine glands to release. So you can see here, you've got your gland, and then right next to your gland, you have the hormone producing epithelial cell, and that's going to go and enter into the bloodstream and be released and be able to flow through, all the different regions of your body because it has access to your circulatory system. Okay. So that's an example of endocrine glands. Exocrine glands, like I said, are slightly different. They have the secretory products that are being released onto the surface of cells. So they can be on the empty surface of a covering or a lining epithelium. So the skin surface and the lumen of a hollow organ, such as your stomach, is a great example of the exocrine glands. So sweat, oil, earwax, wax are all types of secretions that end up on your skin. We'll talk about those more next week, actually, when we're talking about the integumentary system. So this is a great introduction to that. And then digestive glands, such as the salivary glands and pancreas. So we'll talk about the different shapes of these glands here in the coming slides, and kind of how those different shapes can structure, function, aid in the release of the different secretions for them. They, produce substances such as sweat to lower body temperature, oil, earwax, saliva, digestive enzymes. Some of these are going to be protective for the outer layer of our skin, and then things like digestive enzymes are going to be highly acidic types of enzymes. Enzymes that are able to break down larger macromolecules that we consume. So we can see the structure here if we're looking at the secretory portion of a sweat gland. Again, skin or the integumentary system is present here, and you can see a cross-sectional view of the lumen of a sweat gland. So these are, again, cuboidal, and you can see that there are multiple layers here, and the layer closest here is attached to the basement membrane. I guess it's worth kind of noting here, and this is kind of buried into this lecture, but if you read the chapter, they talk about kind of if you were interested in tissues a ton and you wanted to kind of if you wanted to study tissues, the study of tissues, and if you wanted to take beautiful pictures as the one seen here, this is called histology. Histology is the slicing and imaging of tissues, and there's different stains that you can throw onto these tissues, and it's generally a pathologist. The pathologist is the person that's going to actually do the histology. So in the case of having a biopsy done, biopsy is a sample of tissue. Often biopsies are done to see if cells are cancerous, and so a pathologist would, well, they wouldn't take the sample, but pathologist would slice the biopsy and do histology to test and see if that was cancerous. So just kind of I guess I buried the lead there, but if you're interested in tissues, that's a great future profession for you. Okay, so structure of glandular epithelium. So still talking about epithelium here. And for epithelium, you can have these unicellular goblet cells, and we'll talk about the functions of goblet cells here in a second. Or they can be multicellular, so composed of many cells that form a microscopic structure or a macroscopic organ. So sweat glands, oil glands, and salivary glands are all examples of glandular epithelium. So these are all types of epithelium that form a gland and create some type of secretion. So secretion, again, is one of the many functions of epithelial cells. Okay, so these multicellular exocrine glands. So I want to make sure I give you examples of each one of these. So these are different types of exocrine glands and different structures. So man, if we're not going through a lot of structure function stuff here, right? I mean, we talked about the different, arrangement and cell shapes of epithelial tissues, and now we're going to talk about different shapes of exocrine glands that release different excretions. So the simple tubular gland here, you can see has a one single bulb structure at the secretory portion that you see here. An example of that would be glands in the large intestine. Here you can see simple branch tubular. There are multiple branches located here. It's just not one single bulb that falls down. And that would be an example of that would be the gastric glands. Gastric, maybe I should be writing this up here. Gastric, okay. And then next you've got simple coiled tubular. Luckily, this is again, the nomenclature kind of gives rise to what that shape actually looks like. Sweat glands are actually of this shape here, this simple coiled tubular. This next structure you can see here is a simple acnar. You can see it's a little bit more bulbular at the bottom. It's rounded, it's unbranched. So glands of the penile urethra are going to have this simple ocular shape. Next here you can see the simple branched ocular. Again, these are a little bit more rounded at the bottom because I mean, someone could make the argument that they look very similar to the simple branched tubular, but they're not so much tube, they're a little bit more bulb. And because of that, you see the nomenclature change. And the sebaceous glands are going to be sebaceous glands are going to be an example of the simple branched acnar. If we look at compound tubular, compound meaning that it's not just each branch is not just a single branch, there are branches upon branches in the compound tubular. So the example that's given here is the bulbourethral gland that is found within the urethra. So you've got these branches upon branches that occur. The compound acnar, the example that we see here is the mammillary gland. So you can see that you've got branches upon branches, but you've got the more rounded bulbous shape on the end of those, which make it the compound acnar. And then lastly, the compound tubuloacnar. So you've got branches upon branches with multiple branches kind of extend in here. And hopefully some of you have kind of probably figured out the acnar glands of the pancreas are going to have this compound tubuloacnar shape. So when you're talking about exocrine glands, they can either be single or branched, and then they can even be compound as well, and they either be tubular or acnar. So you can see that when people are describing these things, they generally try to almost use two ways to describe them: shape and number, shape and arrangement. So again, maybe just go through this slide, and all of the examples that I just gave you are also located in your textbook. So maybe just go through the slide and list out those examples if you didn't get a chance to catch them while I was going over them here. Okay, so functional classification of glandular epithelium. You can see here that there are different ways in which secretions occur. So the example of the salivary gland is a Merrick-Krins secretion. The Merrick-Krins secretion you can see here. You've got your ER, and it's sending that protein secretion to the Golgi, and that Golgi is releasing it through the vesicular transport process that we talked about in chapter 3, and it's just secreting, causing exocytosis. It's secreting that to the extracellular environment. So it's kind of just straight up normal exocytosis of which we talked about before. So it's fusing with the membrane and then releasing that secretion to the outermost environment, and the original cell stays intact. So it's probably worth noting that the cell stays intact. In the case of mammillary glands, When the secretion occurs, this is called apocrine secretion, and this is where kind of a large part of the cell kind of buds off and bursts. And so this is how essentially milk fats are released from the mammillary gland. And so it's kind of a larger cell portion that gets released here, portion that gets released. And so you've got all these milk proteins that are created from the cell. Again, you've got your Golgi complex here creating all of these milk proteins, and it's going to just basically be released from the cell and cell lyse and be released into the outer extracellular environment. Next, if we look at sebaceous glands, this is called holocrine secretion. This type of secretion occurs via a lot of cell lysis. So the cells essentially just detonate, lyse themselves, explode, and you've got the mature cell is going to die and become the secretory product. And so there's a lot of mitosis that occurs here, a lot of cell turnover. Cells are dying, they're being replaced because the cells on the outermost surface are essentially just almost like blowing up and releasing those that secretory product to the outside environment. So these are the three different types of secretion kind of methods that can occur when we're talking about glandular epithelium. So merocrine secretion, which is exocytosis, apocrine secretion, which is kind of a pinching off of a large part of the cell, and then that's being released to the extracellular environment. And then holocrine secretion, which is essentially cell lysis and release of the cell secretion into the extracellular environment versus that method. Okay, merocrine, apocrine, holocrine. Salivary, mammillary, skin. Okay, so that's all I have for you today. Please make sure that you watch this, you go over those study guide questions for chapter 4. Like I said, we didn't go over membranes in this lecture, so that will be covered in part 2. So make sure you're not worried about finding that information for Tuesday. Quiz 4, chapter 4 is due next Friday at midnight. And then homework 5 is adaptive practice chapter 5, and that's due the following Monday. This is all posted onto the Moodle. It reminds you, kind of gives you an update of your due dates on the Wiley Plus site. So if you're logging onto that regularly, you'll see that as well. And if you have any questions or concerns about this material, please feel free to email me and reach out. And I hope you all have a wonderful day. Thank you so much.